So thank you, Jill, and it's my pleasure today to be presenting on behalf of SPARC K-Core and MapCore on something that we call SCKAN, the SPARC Connectivity Knowledge Base of the ANS. And I invited several people from outside of SPARC, so I thought that I would start with an overview of SPARC and its SPARC infrastructure in order to situate the discussion of the knowledge base in the larger SPARC program. So, as many of you on this call know, SPARC stands for Stimulating Peripheral Activity to Relieve Conditions. The goal of this program is to increase our knowledge of the anatomy and function of the autonomic nervous system for the purposes of being able to develop more effective and targeted neuromodulation therapies. So our approach is to be able to deliver detailed, integrated, functional, and anatomical maps and models of the peripheral nervous system and its innervation of target organs. SPARC is a large project. It can be accessed through the SPARC Portal, which was launched in March of 2020. And this slide is out of date, but there are many, many research groups that span over 100 institutions and companies and that are working across over 15 organs and systems. And it is the role of the SPARC Data and Resource Center to take all of this hard work that's going on and organize it and make it presentable and useful to the scientific community at large. So the SPARC DRC itself is organized into several cores. There's the DAT core, which is run by Joost Wagner at University of Pennsylvania. And that is the data platform where investigators deposit their data and where it is curated, organized, and made available to the public. There is also the K-Core, which I co-lead with Bernard de Bono, which is responsible for knowledge management and curation services. The Map Core, which is producing some of these very detailed maps and is responsible for synthesizing data using common 3D coordinate systems. And finally, the Sim Core, which is, provides a modeling and simulation platform that is available for researchers to work with SPARC data in order to address some of these questions that are involved in neuromodulation therapies. So I'm going to be talking today mostly about the work that is going on between K-Core in the area of knowledge management of connectivity information and the Map Core, which is taking this and building dynamic maps of ANS circuitry. So just a shout out to the SPARC Portal. The SPARC Portal provides access to all of the products that are being developed by SPARC, and recently we released a faceted search capability so that you can go into different categories of information, click on these, and then use a set of facets to be able to hone down into the type of data or modulation simulations that you're interested in. And the reason I also wanted to show this is that now underlying search of the SPARC Portal is something called the SPARC Knowledge Graph. And the SPARC Knowledge Graph is a product of K-Core, and it essentially is taking all of the data sets that are being produced, models and simulations, it is mapping them into common frameworks and making them available through a single endpoint, which we call the SPARC Knowledge Graph. But part of that knowledge graph is something called the Connectivity Knowledge Base. And that is, we don't want to just be able to explore SPARC data through individual anatomical structures. This is a project about the autonomic nervous system, which of course connects structures and spreads influence. And so the connectivity of the autonomic nervous system is a major organizing feature of the SPARC Portal. So we do this almost all of this by mapping everything that we do to common vocabularies. And a lot of those common vocabularies are anatomical ontologies that make sure that we understand the connections between the anatomical structures. Most of the core anatomical ontologies, however, only connect things through part of relationships. So the ventricle is part of the heart. What we're also adding is a layer where that connectivity is nervous system connectivity. So how is it that the nerves connect all of these different structures? So I wanted to spend a little time going through the SPARC connectivity maps and the current interface and how they work. This is, of course, the purview of MapCore, and Peter Hunter is the PI of MapCore. And if you've explored the SPARC portal under maps, you see these multi-species maps, which have connections. So each of these represents a connection that is going through a nerve. But you also see that there are little points here, and these represent places where SPARC has additional data or simulations or tools. So you can actually explore the SPARC contents through the lens of connectivity. So the question, and I should point out that these are multi-resolution maps, and so if you zoom in, you can see that the connectivity is actually quite complex. And as you zoom into these maps, additional complexity is revealed. So the question then is, how do these maps come into existence? How is it that these connections are put onto these flat maps? Well, these are being populated right now on a system-by-system basis for the most part. That is, we have knowledge engineers and other experts who are creating detailed subway maps of circuitry of individual systems. So for example, you see here a bladder connectivity model that was produced by Janet Keast as part of a collaboration with Bernard de Bono, something called the SPARC Anatomical Working Group and other experts, to really provide these detailed, exquisite maps. So these maps, once they are produced, are transferred over into these connectivity maps that you see available through the SPARC portal. But the current way that this was done, or the way it was done for the first several years of the SPARC project, was a manual process. That is, we would produce these very detailed maps, like you see right here. This is just an SVG version of it. And then that connectivity would get transferred over into the user interface that is provided by SPARC MAPS. And I think so there's one feature that I want to first mention about these maps, because this is going to be important when we discuss the knowledge base. If you look at these individual lines that you see on the maps, these each represent different neuron populations. These are neuron populations that are identified by the expert, in this case, Janet Keast, and they course through nerves. So if you see a SPARC map and zoom up, you see these little cuffs. These little cuffs are the nerves, but the lines that you see running through them, classified according to type, represent neuron populations, sympathetic, parasympathetic, sensory, and motor. Okay, and so when you hover over some of the SPARC map currently, you will get anatomical structures, which you see right here. But if you hover over these lines, you'll get something called Keast bladder neuron type 9. And this is a neuron population that we have labeled according to the name of the model and just an identifier that we assign. So these each represent individual populations that course through the nerve. So that's an organizing principle that I will revisit several times. But I think you can see that there's a lot of limitations to the current manual approach, right? That manual drawing and upkeep leads to real difficulties in these types of representations. And some of those problems are in keeping pace with new connectivity knowledge. So these models are constantly revised, new ones come into existence, some of them maybe need to modify, and that's a very tedious process if you need to do this manually. There are errors that are identified and they need to be corrected. This is an extremely complex system. Representing granular connections and roots is also extremely difficult. So if you look at the way that roots are represented in the flat map as opposed to these subway maps that are being produced by Bernard and anatomical experts, there is a loss of topological information. If you want to try to understand connectivity patterns, right, being able to look at what structures are innervated or not is very difficult to follow through these manual maps. And if you want to compare across species and understand which of these connections come from directly, for example, from the rat, or which ones are inferred from other species, all of those things are very difficult with the current manual system. So we have been working since the inception of SPARC on being able to turn these visual maps, which is usually the way we understand connectivity, through these visual roadmaps and pathways that represent the nervous system, into a comprehensive and, more importantly, computable knowledge base, which can serve as the basis for SPARC queries, SPARC maps, and a host of other visualizations. So that's what I want to focus on for the rest of the talk. And we call this knowledge base SCKAN after many, many different names that we apply to it. And sometimes you'll still see SPARC Connectivity Knowledge Base, but for the purposes now, these are both coextensive. And so the knowledge base is really focused on exactly what I said. It is these detailed connections that connect together anatomical structures. And those things arise from the literature, from expert input, from SPARC data, from ontologies. They come from a variety of sources, but they are all mapped to common vocabularies so that they are fair. And there's different representations of vocabularies we use. Uberon is the general anatomical panspecies ontology, but sometimes supplemented with the foundational model of anatomy or the adult mouse anatomy ontology. These are all interoperable with each other. And they are represented using a formalism called an ApiNATOMY, which I will discuss in just a moment. So the motivation for SCAN is that I just mentioned, right? That we want to move from a manual workflow that takes these and draws them in other contexts to a dynamic system that can support knowledge-based queries. So such queries as find anatomical regions connected by specific neuron population types, find connections that course through a specified nerve tract or ganglia. We want to be able to automatically populate models, interactive visualizations, simulations with up-to-date connectivity information. And we also want to be able to attach all of this information to very detailed and fine-grained provenance. Sometimes one has provenance for an entire neuron population, but more often than not, the fine-grained roots all come from separate studies, and we want to be able to attach that fine-grained provenance to the individual parts of a given neuron population. But there are challenges in building such knowledge models of neuronal connectivity. In order for us to do this effectively, we need to be able to change information that is contained in these visual maps into a knowledge model that also supports certain types of reasoning and other things in order for us to be able to answer those queries. That is, it can't just have those keywords in there, but it needs to be able to traverse the relationships between these to build a coherent knowledge base. And why has it been so difficult in creating knowledge bases for neuronal connectivity? Well, most of the knowledge models that we have assume that a cell is a cuboid thing that exists in one anatomical compartment and no others. But in fact, we know that neurons are complex cells with segments that span multiple anatomical structures. The nerve themselves are not single entities, but there's multiple branches with different names, and different neuron populations travel through one or more of these branches. They have different ingress and egress routes, right? So nerves are not considered coextensive with neuron populations. They rather provide the conduit through which these neuron populations traverse. The trajectories are very complex. And I think that that's really appreciated by the two diagrams that I have down here. The left one is from Gray's Anatomy, and most knowledge models that we see of connectivity in the nervous system are very simple. It's region A projects to region B via nerve C. But instead, when you actually track and try to put some realism into how these neurons actually go, you see it's not linear, and they go through various structures on their way to their final target. So all of these things have presented a real challenge for knowledge representation. And over here, we just take this one neuron population that you see over here, starting in the lumbar spinal cord. And this population alone traverses over 25 different anatomical structures on its way to its target. So these are big challenges for knowledge representation. So to solve this challenge, we are using a tool suite and knowledge representation system that was developed by Bernard de Bono called ApiNATOMY, which literally stands for an API for anatomy. And this is an exquisitely detailed and very elegant model that basically models flow between biological structures. And the flow between biological structures can represent a fluid such as lymph or blood or cerebral spinal fluid, but it can also represent information, which of course is what we are concerned about in the central nervous systems. It allows us to be able to represent neuron populations and the location of their parts. So we can assign location to as many different parts as we wish to assign it: the soma, the dendrite, the axons, the terminals. And the main advantage of ApiNATOMY for the type of work that we are doing is that it solves the chaining problem. So it allows us to be as detailed as we want in these individual segments, but then it provides a compact and computable representation for chaining together the individual segments of these neuron processes so that they can be treated as a whole. And this is the real advance, in my view, of ApiNatomy, is allowing us to do this in a compact way. And as I mentioned, because all anatomical structures are mapped to their appropriate identifiers, it is an infinitely fair model in that it is interoperable with any other system that also maps itself to these same identifiers. So let's just go a little bit into detail in ApiNatomy. Of course, I'm not the expert on this, but Bernard de Bono is. And he is here today if you have any questions. But just at a high level, the ApiNatomy formalism models tissues and their relationships through a series of entities that are called lifts, which can basically be thought of as the tissue material, links, nodes, and chains. And these allow us to define, for example, the location of a cell soma. It can be situated in a larger lift, so this would be one lift, and it can be situated in a larger lift, which might be an anatomical region. And it can also allow us to define the various segments of processes that traverse across different tissue lifts. The chains and the nodes and the links are what allow us to put these together into a coherent whole. So this is, for example, some modeling work that was done by Monique Surlis-Ziegler, Natalia Kokash, Tom Gillespie, and Bernard. And what you see down here is a representation of a complete put together neuron population that has its dendrites in the spinal cord, its axon over here, and then its axon terminals in the bladder dome and the bladder neck. And you can kind of see the little individual components that ApiNatomy defined, which it then appended together to form a single chain that can be used to look at gross connectivity. And as I mentioned before, ApiNatomy, as one of the key things that we need, also allows us to assign evidence for each of these types of biological connections. So each of those segments or any statement that we make, we can actually attach biological connection information to it. As you can imagine, these models are somewhat complex to actually create, and Monique and Tom and Natalia and Bernard deserve a lot of credit for working through initially just spreadsheets and trying to put these models together and turn them into this formal representation. But in order to assist in that process, both in the authoring of these relationships between the different compartments and also for troubleshooting, because you can imagine it's very difficult to troubleshoot. In fact, we have been working on a whole set of authoring tools. It's a visual authoring environment that lets us compose these models and also test them to make sure that the connectivity represents what the expert intended. And we are actually working with a professional software development company called Metacell in order to make these tools available. They are all open source and will be available to the community at large. But this was a very important component in really trying to scale up the representation. So the basic workflow then for populating SCAN with these models here is that these are created through ANS experts, the SPARC Anatomy Working Group, or SOG. They produce this map. They then use the authoring tools and other tools, the knowledge engineers, in order to convert that into something that is computable. And then these are stored in something called SciGraph. SciGraph is a Neo4j-based graph database that lets us actually traverse these relationships. It is available through a SciGraph instance, which is mostly what I'll be talking about today. But for those of you who are knowledge engineers, it's also available as an RDF store via BlazeGraph. And we'll be happy to talk to you about that if necessary. So the current models that we have in SCAN are 1, 2, 3, 4, 5, 6, right? They are continually being added. But the ones that are in SCAN version 1.0, which is our current release, represent the Bolzer-Lewis model of defensive breathing, which comes from Don Bolzer and Steve Lewis, the Keast bladder model, and these are all SPARC investigators, from Janet Keast, bronchomotor control, the Jeff Ardell, John Armour model of the heart, the SOG model of the distal colon, and that primarily came from the SPARC Anatomical Working Group, and the Powley stomach, which is a stomach model that came via Terry Powley. And together, these represent about 91 different neuron populations. And you can see the complexity of the connections in each of these individual SVG diagrams that were produced by these experts working with Bernard de Bono. So if we revisit the type of scan use cases that we were talking about earlier, I wanted to actually go through and show how SCAN is capable of answering these questions. And I'm sorry for the jet plane that is flying overhead. I hope you can't hear it. So essentially, we replace those static drawings with the SCAN knowledge base. And through dynamic queries, we're able to support knowledge-based queries, like I talked about before, find anatomical regions connected by specified neuron types, find connections that course through a specified tract, nerve, or ganglion, and automatically populate models, interactive visualization with up-to-date connectivity information, and then also provide detailed provenance. So I'm going to show these use cases, and forgive the ordering here, but the flow worked better if I show this one first and then the second knowledge-based query last. So the first use case is simply find anatomical regions connected by a neuron population type. That would be one of these things that you see here. It has an axon, a dendrite, a soma, and axon terminals, and it plots a course through one or more structures. Usually more is rather than one. And if you issue that query using a language called Cipher to the SPARC to scan, it will actually produce a list of parts, including an axon chain, which it has put together from the information inside of ApiNATOMY. So in this case, I'm only representing the axon chain, but you can see that for each part, each segment that we have, it gives you the anatomical structure in which it is situated. And if we put those things together, we start to plot the route of these neuron populations, including their enclosing nerves. So here you can see in a very simple visualization, the type of information that can come out of SCAN. Here we see some of the latest work, though, that's going on in visualization. And this looks, of course, like the SPARC map that you would see on the SPARC portal, and it is. Only this time, this neuron here that is drawn in blue was not drawn manually, but it was drawn by issuing a dynamic query to the SPARC knowledge base and then put together so that the course, the accurate course of the nerve reflects the type of information that is in the SPARC knowledge base. And there are a few little rendering artifacts, so you do see some disconnections between the segments. But I think that this is a great advance in SPARC, and I wanted to acknowledge David Nickerson, David Brooks, and Tom Gillespie for putting this into existence. So this particular blue line here was not drawn by a human being, but was drawn by a computer. And it was done by, again, issuing a query to the SPARC knowledge base, which returned the necessary information, including about what parts are connected with what and in what order. And if we look at that in more detail, we can see again that it does represent end terminals, the position of somas, and the routes through the anatomical structures that are traversed by these neurons, to the extent that they are represented in the flat map here. The flat map, of course, doesn't have everything because it can't, it's a 3D visualization, but it is an accurate reflection of the route that has been specified in ApiNATOMY. So I think that this is a major advance in SPARC, that we are now able to start populating our connectivity interfaces via the SPARC knowledge base instead of through manual entry. So as I mentioned, we're also able to attach provenance. And so when one starts to look through then these individual neuron populations, you will be able to click and it will tell you where that information came from. And it will tell you where that information came from both at the model level. So each of the models that are produced by our experts are published in Zenodo, and they have a DOI, or they will be published in Zenodo. This is the KEIST model. So in every case, it will tell you which model it came from, but it will also provide a list of sources of literature that will be used, that you can use to explore those connections further. This is currently in beta testing. It's not available through SPARC just yet. And right now we're really doing this at the neuron population level, but as I indicated, the scan itself contains very granular information to the extent that we are provided that by the experts for very specific parts of the connections. So the last use case is find all neurons with processes in a given structure. In this case, it's the inferior mesenteric ganglion. And this really relies on the fact that even though these models were all produced independently by different experts and Bernard, They are all mapped to common reference ontologies. So anytime the IMG appears in the model, it appears under the same identifier. So it doesn't matter that in one model it's called the inferior mesencephalic ganglia, and in another model it is called the IMG, because it's mapped to the same identifier. The computer knows that it can actually put that information together. So this is what we mean by making data fair. It is interoperable in this case. The I in FAIR stands for interoperable because it is all mapped to a common model and a common formalism. So if we issue a cipher query to SciGraph and we say, find all neurons with processes in the IMG, it returns a list of neurons from whatever model has information that goes into the IMG. In this way, for example, models that come out of the bladder can be combined with information that come out of the colon because they're referencing a common set of structures instead of these things all being in isolation. And this is the list of neuron populations that it returns. You can actually represent connectivity in SCAN as a matrix. And on the left here, you see all the neuron populations that we have. And underneath here, you see all of the anatomical structures we have. And essentially, it's just a lookup. Here's the inferior mesencephalic ganglion, and I'm not expecting that you would be able to read that. And it says, here are, I think, 10 neuron populations that have processes in there. And these can be further color-coded, as I'll show you, in terms of the type of process there, but these are just the neuron populations that course through this particular structure. So here you can see that there are two models that are returned. One is the colon, and the other is the bladder model. Here's the IMG here, here's the IMG here, okay? And I've color-coded the neurons that come from the colon model versus those that come from the bladder model. If we look at those a little bit more detailed, as I mentioned, it's not just that, that those neurons course through, but we have some idea of the neuron parts that are involved. So over here, I have my imaginary electrode that if one stuck into the IMG, you would be hitting the somas of sympathetic postganglionic neurons. You'd be hitting dendrites of sympathetic postganglionic neurons, axons, somas, axon chains, and so on down the line. So it's actually giving you that information at that level of granularity. So if we actually then plot, for example, all of the connections that we see for the IMG, we see that there are 10 neuron populations, but that these traverse 30 anatomical compartments, and these are all listed right here. I very clumsily tried to map those onto the flat map by these circles, and you can see the sphere of influence of potentially structures that might be impacted by stimulation. Granted, they're not all axons, but some, that there's still always a potential when you put an electrode in to evoke a response. And I think you can see how powerful this might be when we're trying to do models and simulations of bioelectronic medicine, right? That you would be able to have some understanding of the sphere of influence at any point along these ApiNATOMY models that might be influenced by a given stimulation protocol, okay? So I think, right, that basically shows you the 4 use cases and the types of things that actually can come out of ApiNATOMY, the SCAN knowledge base. The model that I just showed you, the route along all of those structures that are influenced by putting an electrode, an imaginary electrode into the IMG, is of course very crude. And in any given visualization, you may run up against a limitation in how much of the detail that is contained in SCAN in any, any particular interface. But the beauty of having it all in a single knowledge base and a single knowledge model is that, in fact, you are not limited to a single visualization. You can drive any sort of visualization that takes advantage of this information in order to be able to show it in different ways. So the boxes that are in purple here are some of the visualizations that are currently available through SPARC. So of course you can look at it as a list of structures. You can look at it as a connected neuron population. This one is unlabeled. This one actually has all of the structures. This is something called a 2-map, which is an abstract representation of the body that lets you look at connectivity across multiple structures without worrying about organs getting in the way. And of course, you saw the flat map already, that it can be visualized on the SPARC interface. You can look at it as a matrix as well. And so I really do believe that these types of knowledge bases, right, these are the things that we ourselves have come up with, but that they do lay the foundation for connectivity atlases that can be driven off of the database, 3D models, as I'll talk about in a minute, and also producing those detailed wiring diagrams, those subway maps, those two-dimensional wiring diagrams that you've seen. All of those, in theory, can be run off of the knowledge base, and we're actively working on some of those. But I do want to bring back, right, the power of the knowledge base for the SPARC project, because as I mentioned before, there are modeling and simulation components to SPARC that will allow people to take SPARC data sets and also knowledge that are contained in the SPARC knowledge base and actually perform in silico biology on this information. And so the models that I showed are the two-dimensional flat maps, but in fact at Auckland, they are working on three-dimensional scaffolds where the organs and other, their individual organs, they can be placed in body parts. And these can be used as the basis of modeling efforts and also simulations. And if you can kind of see this little gold line here, this actually represents the vagus nerve. And so you would be able to go to the knowledge base and actually populate a three-dimensional model with neuron populations that actually start to course through the vagus nerve and connect up these organs. I also want to give a shout out to the OSPARC platform that is part of SimCore, and this is being run by Ezra Newfield of IDIS. They have also very powerful 3D models. What you see over here is a skeleton that has the blood vessels and also the nervous system represented. And again, to the extent that we are all using common anatomical ontologies, you would be able to populate these neurons or nerve paths with information that come from SCAN. And this again is a computational simulation platform. So you would be able to simulate stimulation and other sorts of things within this platform. So now that the knowledge base is up and running and populated, we're expecting to see that information is now going to be making its way into these more computational environments so that it can be acted upon. Okay. So as you can imagine, trying to keep something like SCAN up to date, being able to add to it, and also augmenting quality control processes is in fact a giant challenge. And we don't want this to just be something that is static. We want it to continually update, and that is being done through the SPARC project, but that can also be done through other routes as well. So I wanted to talk a little bit about some work we're doing using natural language processing to help us add additional information, maintain the quality of the database and integrity of the database, and also implement some QC processes to make sure that the information that we have is accurate. Okay. So I mentioned already one route by which SCAN is being populated, and that is through the ApiNATOMY route with these very, very detailed subway maps, as I call them, authored through ApiNATOMY and put into SciGraph. But we've recently implemented another route, which involves using natural language processing to digest the literature and look for connectivity statements, where we see evidence that a connection that is of relevance to the autonomic nervous system is being mentioned. These statements, as I'll show you, are then vetted by curators and experts, and they are modeled according to something called the Neuron Phenotype Ontology, which was developed by Tom Gillespie. There is an article that is in Neuroinformatics, which is in press on this, and also a preprint in bioarchives. And these also make their way into a common store. So I wanted to spend a few minutes talking a little bit about this pipeline because I think it has a very important role to play in SCAN. So essentially, we trained a classifier, and this is the work of Barak Ozert and Joe Menke in my lab at UCSD. And we trained a classifier to be able to recognize when two anatomical structures were mentioned in a sentence and where there was a connection between them, okay? And we divided these types of classifiers into recognizing functional connections, where it is a physiological connection, anatomical connections, something that we call structural. For example, nerve A, branch A is contiguous with nerve branch B. topological that represents, for example, a course of a nerve through a given structure, and then just what we call general connectivity, where it's very hard to tell exactly what type of connectivity is being referred to. So we developed an NLP pipeline that would recognize these things. We trained it, and we deployed that over the open access subset of PubMed Central, which is currently about 2.5 million articles. We were able to obtain a lot of these different types of connectivity statements. We, as I'll show, we use a curator to be able to track them down. But once we get these anatomical statements, we model them using a much simpler formalism than we do for ApiNatomy, because oftentimes these are fairly generic and we don't have detailed information about their individual roots. So as I mentioned, we're using something called the neurophenotype ontology. It represents this information in a language called OWL. And it's just very simple. It's that structure A projects to structure B via nerve C. So here you have the pteropalatine ganglion projecting to the lacrimal gland via the greater petrosal nerve. And for those of you who are interested in knowledge representation, this is the way it is represented as a set of OWL restrictions, again, referencing the same set of common identifiers that we're using in the ApiNatomy model. So currently we have about 90 neuron populations that have been added to NPO, largely covering the cranial nerves, in order to sort of augment the information that we have via ApiNatomy. And I probably should have shown this slide first, but somehow it got out of order. Essentially, the way that we use it is that we run the NLP pipeline. It gives us a set of suggestions. It finds the sentences where connectivity information is there. That goes through a curatorial review process. And part of that curatorial review process is, does it exist already in an ApiNatomy model? Does it exist on the flatmap? And if so, is it consistent with what is currently represented? Is it not consistent? And you can see over here an example of one of the sentences that was pulled out. Phrenic motor neurons innervating the diaphragm are located in the ventral horns of the lower cervical spinal segments and receive bilateral excitatory synaptic drive from ramp 1 and late E neurons. But when we actually look on our flat map, we see that we have the phrenic motor neurons going to C4 and C5, and this is saying the lower cervical spinal cord. So a curator would go and look at the primary references, try to figure out what it is that this is saying, and then compare it to the information if we have it. And one of the things we're working on is the knowledge model for how to deal with conflicts, right? That we might have differing information coming from our models, our experts, and the literature, okay? So everything doesn't go directly from NLP into the knowledge base, but there's always this process of curatorial review. Once those statements have been determined to be accurate, we undergo the knowledge engineering, like you saw before, turning those into OWLs, and they're represented in the NPO, and then they go into the SCKAN knowledge base. So why are we using this? Well, we're using it for several purposes. One, as you could see, to ensure that connectivity within SCKAN reflects the current state of knowledge, and make sure that what we have, right, if there are conflicts or species differences, that we can identify them, right? So we are looking for statements that either contradict what we have, but sometimes they just augment them. They provide additional evidence, and we would like to add that to the provenance. Species differences right now, if you look closely at the SPARC connectivity map, it's the same map superimposed on different species, but we know that there are species differences, and we need to identify those and make sure that we have those in the knowledge base and available for query. We want to be able to keep up to date with new knowledge, so the pipelines run every quarter so that we can see if new information has come in, new work has been published. We want to actually be able to compare functional connectivity to the current state of knowledge. So if you look at a lot of papers, they will talk about connections between two structures, but you realize they're talking about functional connectivity. And we're kind of curious as to whether we can map anatomical roots that would account for that functional connectivity. And as we move forward, we'd also like to be able to augment the type of connectivity knowledge we have now with additional depth. For example, you see that we have these anonymously named neuron populations, but some of those correspond to individual neuron types that are well recognized, and we would like to be able to add that to the knowledge base. And also, as you see here, we have molecular constituents that are associated with these pathways, channel receptors, all kinds of things that we could layer on top of it in order to provide added depth to the knowledge base. So we think that this pipeline is going to help us to do that because it doesn't involve humans having to read the entire literature, but we get an assist from the computer. And as the models get better, we expect that we will be able to employ less and less curation as we move forward in order to make use of this information. But right now, it is an assisted pipeline that involves both humans and computers. Okay. So I wanted to finish up with a little bit about how you can access SCAN. So right now, the first release of SCAN was done in August. And if you want to query SCAN yourself, then you need to know Cipher or Sparkle. So that is certainly beyond what I can do. But Tom can help you with that if you have any interest in particular types of queries. We did create, there is no programmatic interface to SCAN right now, except in a very sort of limited way. And that is mostly because of the computational burden of doing some of these queries. But we do have a Dockerized release of SCAN that is available right now. It's described in Zenodo. It has its DOI right here. So if you wanted to install it yourself, we've included some example queries that are available through one of our pages on the SOG. And you can answer queries like the two that I showed you. And this one, in fact, you can play with yourself because you can paste that in your web browser and just change the number of the neuron here. So you can look at all of the models that are available with neurons that are available with the Keys model. But for the other one, the Find All Neurons one, that's a much more computationally intensive query and requires that you install SCAN. But we provide detailed documentation on how to do that. But where is SCAN going? So as I said, we've already released the first version of SCAN in September 2021, and those will be released quarterly. So we have another one due in January. I mentioned a lot of the tools here that are currently under development. That is, you won't see them available through the SPARC portal. The next version will include the pancreas and the spleen. The detailed ApiNATOMY models have been created for those. So those will be included in the January release. That visualization of SCAN using the SPARC flatmaps and other tools, that will be released in a future release of the map part of the SPARC portal. But right now, it's still in alpha stage. The full suite of authoring and visualization tools for constructing ApiNATOMY models are available as open source software, so you can contact us if you're interested. Those are still not fully developed though, and they will be released in the near future. Some of them, I think, with the January release. I should mention that we have both information from the NLP pipeline and ApiNATOMY in the SCAN database, but currently you can't query them as a unit. So we are currently working on a unified query strategy so that you can pull out both information. Also, we're working on integration of SCAN, ApiNATOMY, and anatomy expert groups into something called the Common Fund Data Ecosystem, and I'll talk a little bit about that in a moment. Planned in the future, though, is that we do want to have a query interface that's integrated into the SPARC flat map so that people such as myself who don't write either Cipher or Sparkle will be able to issue basic queries of the type that I showed you and be able to return answers. As I said, we're working on the handling of species difference, contradictory evidence, and controversies, and those will be released in a future release. So I just want to give a shout out to the CFDE. So this is an umbrella organization for all of the big projects like SPARC that have been funded through the Common Fund and are generating large amounts of data. Bernard de Bono in particularly has been taking this very unique asset that we have, which is SCAN, right? Nobody else is dealing with connectivity. There's a lot of molecular mapping and physiological work, and we want to make SCAN available to the Common Fund data ecosystem so that all projects can look at patterns in their data through the lens of connectivity. So he has taken the concepts and some of the infrastructure that have been developed by SPARC for populating and vetting the connectivity information in the form of the Anatomical Working Group and ported those over to the CFDE. And so SCAN and ApiNATOMY are being used to, in fact, manage anatomical knowledge across different projects such as HubMap, Clovac, and ReproTox, and those tools should be coming out in the next year. So looking forward.
So going back to that slide, Jeff Ardell asked a question in the chat, which asks, will there be a pathway for SPARC investigators to add additional?
I can answer. Yes, there will. So let me, I should say that part of the ApiNatomy tool suite is something called OSCAP, which should allow investigators to be able to interact with the maps much more directly. So thank you for bringing that up. Okay. So anyway, this was my big finale, Anita, and then I was going to answer questions. So looking forward, I wanted to really try to convey some of the excitement that I have over the work that has been accomplished by Tom and Bernard, Andre, and others in constructing the knowledge base. I got into informatics back in the 1990s, and what you see over here, for those of you who are a bit older, know that this is one of the most famous diagrams in neuroscience. This is the Fellman and David Van Essen model of connectivity in the visual cortex. And it has been a long goal of mine and many others in neuroinformatics that we take these out of static diagrams and turn them into something that is computable and interoperable, in other words, FAIR. We are generating tons of data through Brain, SPARC, and other projects that deal with multi-scale connectivity. And I really do believe that the type of work that we are doing is going to lead to, as I said, next generation's atlases, interoperable circuit diagrams. Imagine if every figure that is published in a paper, instead of being static, in fact, was driven off of something like SCAN so that they could be compared, rearranged, and queried. So I believe that a long-time goal of neuroinformatics, which is to be able to build a multi-scale knowledge base of known neural connections and circuits, is actually feasible. And I think such a thing has great use outside of just basic neuroscience for electroceuticals, as we've talked about, brain-machine interfaces, and also being able to understand and model things like disease mechanisms using the information that we have. So I consider that, if we could do it, to be equivalent to GenBank or Uniprot or some of these very large databases, which represent the sum total of a type of entity. In this case, our entity are connections. So with that, I will end the talk and just give a thank you again to everybody whom I mentioned on the slides and some that I didn't, but I think you can appreciate that this really was a large amount of engineering work. Can't claim that I did any of it, but it was my privilege to actually oversee it and also present it, but it's really the work of Bernard and Tom and David Masterson and Barak trying to bring these things into existence. So thank you. I think we have 10 minutes for questions, if there are any.
Sorry for stopping you before, Marianne.
Yeah,
I just thought that that slide was actually right there, because one of the big outcomes of this is not just to drive the SPARC funding towards this kind of a goal, but also there's now a larger group with a common fund that is also going to be taking advantage of and adding to this important knowledge base. So
it was this one here. Okay. Yes. So to your question, Jeff, is right now the tools are directed towards people who are I should say dedicated, right? That is, right, somebody just sort of casually walking by would not necessarily be able to do this. But one of the things Bernard and KCOR has been working towards is to at least lower the barrier to entry to SPARC investigators and those who really want to represent accurately the type of connectivity information that they're generating. And I think the new generation of tools is going to allow that to happen, especially the OSCAP interface that Bernard has planned. So if that is something that you're interested in, we're looking for users to help spec out exactly how that might work, and we will be in touch.
Yeah, well, we're certainly interested, but we're also, like you said, we're also very much involved in already implementing very targeted neuromodulations, which engage these populations to see what effect it has. So there's obviously a huge potential here.
Okay. Yes, so we would love to follow up with you on that. Other questions, comments?
To what extent do you also store the raw, maybe image data sets or other data sets that were used to produce the maps. So I know you said you can link to the publications and original sources, but are there direct ways to query the original data sources as well, or is it still kind of indirect?
Well, to the extent that, so any of it, especially with the expert maps, a lot of those are coming from data that they are actively generating and making available through SPARC. And so currently that connection is rather crude, but in the next phase of SPARC, we're planning on making that much more granular, right? So that if a connection came from a SPARC data set, you would be able to visualize it. And the SPARC portal, of course, has a bunch of tools that allow you to do that. But we don't always have the control over what comes from the materials in the literature. If they do make their data available, it would not be difficult for us, if it's still in existence, because a lot of these papers are old. If they, if it's well managed and curated data, it would not be difficult for us to also add links if it is stored in an archive someplace that we can access. So, that would be actually not something that I had thought about, but it would be a good addition, I think.
Cool, thanks.
Okay, I think if we don't have any more questions for Marianne, we'd like to say thank you. That was an excellent talk. It's always fun for those of us in K-Core, because we're always in the weeds and trying to put everything together. So it's great when Marianne presents it all in the big picture, like for all of us. So,
Yeah,
thank you.
well, I hope it was understandable, because it's a lot of complex representation. Yes. But anyway, it was my pleasure to present on behalf of the group. Yes.